Assalamu alaikum and hi everyone. This video is about the monosaccharide derivatives and oligosaccharides. The content will be more focusing on the monosaccharide derivatives than the oligosaccharide. Learning outcomes for this topic are, at the end of the video, you should be able to 1. Differentiate the different types of monosaccharide derivatives. 2. You should be able to explain the oligosaccharide sugars, and lastly, you should be able to describe the function of these two compounds. Before we go further explore the topic of this video, it's good to have a quick revision on the main carbohydrate classification. In general, carbohydrates can be classified into three major groups, which includes monosaccharides, oligosaccharides, and polysaccharides. Monosaccharide is the basic unit of carbohydrates that consists of only one unit simple sugar. The detailed classification and naming of this sugar have been discussed in the previous video. Oligosaccharide is sugar that have between 2 to 10 units of monosaccharides. It can be named according to the number of monosaccharide unit it has. For example, disaccharide is a 2-unit monosaccharide sugar, and trisaccharide is a 3-unit monosaccharide sugar. For polysaccharide, it is a sugar with more than 10 units of monosaccharides, which can be classified into homopolysaccharides or heteropolysaccharides groups. Carbohydrate can be formed into its simpler or more complex form through two important reactions, whereby a breakdown of complex sugar into simple sugar can be made through hydrolysis reaction, where water is consumed. Meanwhile, formation of complex sugar from simple sugar can be done through condensation reaction that will release water from the compound. Next, we will move into our main content. Because of monosaccharide are abundant with hydroxyl groups, sugar molecules can form several types of important derivatives, including sugar acids, sugar alcohols, deoxy sugars, amino sugars, and glycosides. Monosaccharide can be selectively oxidized at the anomeric and primary hydroxyl group. The oxidative reaction will produce a compound named as sugar acid. The oxidation produces carboxyl group COOH at one or both ends of the structure through chemical or enzymatic oxidation. Main classes of aldose sugar acids are aldonic acid if oxidation occurs at C1, uronic acid if oxidation occurs at C6, and alderic acid if oxidation occurs at both ends C1 and C6. For example, glucose will produce gluconic acid, glucuronic acid, and glucaric acid if oxidation occurs at the respective end as shown in the figures. Due to the properties of sugar can be selectively oxidized, carbohydrates that can reduce oxidizing agents are referred as reducing sugar. All common monosaccharides are reducing sugars. The common disaccharides maltose and lactose are reducing sugar. However, sucrose is a non-reducing sugar. The clinical significance of reducing sugar properties can be used to detect urine glucose in diabetes mellitus condition and to detect fecal reducing substance to diagnose carbohydrate malabsorption such as lactose intolerance. The presence of reducing sugar can be detected through simple qualitative tests by detecting color changes in the indicator solution. The common indicator solution used are the oxidizing agents such as Benedict's solution and Ferling solution. Both consist of copper sulfate, which diluted in different solutions of alkaline citrate and alkaline tartrate respectively. Reducing sugar will reduce copper to ions, the deep blue color solution, to copper-1 ions the brick red precipitate of copper-1 oxide. The second monosaccharide derivatives we're going to cover is sugar alcohol. It is also known as alditols or polyols for having multiple hydroxyl groups. It derives from reduction reaction of monosaccharides carbonyl group. It is water-soluble solids which can naturally occur or industrially produce. Sugar alcohols can be found naturally in plant products such as berries and fruits. It has the characteristic of sweet tasting, which can be labeled as sugar-free, which contain less than 5 grams of sugar per serving, or no sugar added means no sugar added to the food manually. However, it still does influence the blood sugar level, but much lesser than sucrose. This is due to they have low glycemic index, which being absorbed at 50% of the rate of the glucose. Few examples of important sugar alcohols includes sorbitol, inositol, and mannitol. Next is deoxysugar. It is the derivatives that consist one or more hydroxyl groups of the pyranose or furanous ring replaced by the hydrogen. A well-known example is 2-deoxyribose, which is a component of deoxyribonucleotides, the repeating units of DNA. It is the derivative that have hydroxyl group at carbon 2, which being replaced by hydrogen. Another example is hydroxyl being replaced at carbon number 6 of pyranose ring, such as L-fucose, a constituent of cell membrane glycoproteins and glycolipids, and L-rhamnose, a constituent of lipopolysaccharides of outer membrane of gram-negative bacteria. The second last derivatives of this topic is amino sugar. Amino sugar is obtained by replacing hydroxyl group of a monosaccharide by an amino group. The amino group usually occurs as the N-acetyl derivatives. The most common amino sugars are the D-glucosamine, which can be found as the main component of chitin of crustaceans and insects, and the other one is the D-galactosamine, which present in chondroitin of cartilage, bone, and tendons. The other example of amino sugar is mannose, which is the constituent of glycoproteins. And lastly, we're going to cover the glycoside. It is the sugar bonded to another group on its anomeric carbon via either carbon, oxygen, nitrogen, or sulfur glycosidic bond. Glycosides are formed when the hydroxyl group of the anomeric carbon of monosaccharide undergoes condensation with hydroxyl group of a second molecule, and this will eliminate water. The sugar group known as glycone and the non-carbohydrate group is called a glycant. Examples of glycosides are fluorescein, digitonin, and streptomycin. The function of monosaccharide derivatives can be described based on the structure or component they are belong to. Mannitol is an osmotic diuretic which used to reduce intracranial tension by forced diuresis. Glycerol can act as the humectant, sweetener, solvent, and backbone of lipid. The very well-known deoxyribose is the component of DNA that carries genetic information in all living things. Other than that, monosaccharide derivatives of glucosamine can act as lubricant in joint cavities, act as the anticoagulant, and is the structural basis of insects. Galactosamine is the constituent of chondroitin, which provide resistance to compression. Last but not least, mannose is part of the component cell membrane and receptor. Next, the second major class of carbohydrates, the oligosaccharide. Oligo means few and saccharide means sugar. Oligosaccharide can be described as carbohydrate with 2 to 10 monosaccharides that are linked together. The most common one is the disaccharides, which consists of 2 units of monosaccharides. It can be formed through condensation reaction of monosaccharide unit and joined together through the O-glycosidic bond. The important disaccharides that commonly discussed are the lactose, which made from galactose and glucose. These biomolecules can usually be found in milk but otherwise does not occur in nature. The non-reducing sugar sucrose is a disaccharide sugar made of glucose and fructose, which commonly found abundant as sugarcane. Glucose can form glycosidic bond with itself and produce important disaccharide known as maltose, which can be found in malt. Functions of disaccharide are explained based on the common sugar only. Sucrose, because of its sweet taste, it is largely consumed as sweetening agent. Sucrose is sweeter than other common disaccharides due to the sweetness of fructose. Lactose is used in infant formulations, as the medium for penicillin production, and as a diluent in pharmaceutical industry. Lastly, maltose can be used as a nutrient, sweetener, as well as the fermentative reagent. That's the end of this topic. Thank you for watching. See you in the next video.